Today, we're almost 7.9 billion people sharing this planet and all of its resources. The population is consistently growing, leading to a constant expansion of integrated agriculture. As a result, the global need for freshwater is greater than ever, and it will only keep expanding. Farming alone accounts for almost 70% of the water withdrawals, putting major stress on the freshwater resources at our disposal. Because despite that 70% of the world's surface is covered in water, only 2.5% of it is freshwater, and of that, as little as 1% is available to us, whereas the remaining amount is found in glaciers and snowfields. Due to this, lack of freshwater is one of the greatest issues we as a population are facing. The effects of freshwater shortage are already apparent in all parts of the world. It's vastly affecting the agricultural prospects in developing countries. But even here in Sweden, the agricultural industry is experiencing hardships caused by the lack of freshwater. It's something that will continue to have a considerable impact on the agricultural industry and thereby all of mankind. To manage this crisis, various methods for desalinating seawater have been developed, and today many areas and industries are dependent on those methods for their freshwater source. However, while these methods have found innovative approaches to desalinate seawater, they have some apparent drawbacks. They're highly energy consuming, and the energy that they use is often not renewable, causing them to leave environmental footprints. Their high energy consumption also makes them very costly, leading to them only being accessible to wealthier countries and industries. To offer a more environmentally friendly and economically accessible solution, we would like to present our project, CyaSalt.
Our desalination solution follows what we have chosen to call the CyaSalt cycle. The CyaSalt cycle shows the four main phases of our method, starting with the cultivation of the modified organisms, moving on to the actual desalination process, and then the separation of the modified organisms from the desalinated water. The cycle ends with part of the biomass being used for inoculation of the preceding cultivation, and the rest being used for biofuel production. The phototrophs we have chosen to work with in this project are cyanobacteria, more specifically the cyanobacteria Cyanocystis sp. pcc 6803. The main reason for us choosing Cyanocystis is that it is a well-studied phototrophic organism that is both halophilic and photosynthetic. This means that they can grow at high salt concentrations and that they utilize sunlight as their primary energy source. Due to these properties, both cultivation and desalination can take place at seawater salinity levels, and they can be driven by sunlight. After cultivating these modified phototrophs, it's time to introduce them to the seawater so that they can desalinate it. And what we mean by desalinating seawater is that the extracellular sodium chloride is transported into the cells and thereby being removed from the surrounding water. The next step in the CyaSalt cycle is to separate the modified phototrophs from the desalinated water. This will be achieved by yet another modification that enables the organisms to be separated from the desalinated water by binding to a cellulose filter. Now we can move on to the last part of the CyaSalt cycle. In addition to receiving desalinated water, we also aspire to make use of the leftover biomass that is stuck to the filter. Part of it will be used for inoculation of future cultures, and the rest can be used for biofuel production. And this completes the CyaSalt cycle.
Now it's time for a more thorough explanation of the cycle. But before we talk about the cultivation part, let's skip to the desalination part and explain how our desalination system works. So, what we want to achieve is a system that enables phototrophs to import extracellular sodium and chloride ions from the surrounding water. To accomplish this, we designed 4 parts that are introduced to the phototrophic organisms. The first part is the inward-directed chloride pump halorhodopsin. Halorhodopsin is activated by light, and when activated, it transports chloride ions into the cells against the concentration gradient. It has been used for the purpose of desalinating water before by a Shanghai iGEM team in 2015. The promising results that they obtained encouraged us to include it in our system as well, but together with 3 additional parts. The second part in our system is the light-activated cation channel rhodopsin. The influx of negatively charged chloride ions created by halorhodopsin generates a negative membrane potential. This, in combination with light, is believed to induce an influx of sodium ions through channel rhodopsin. When researching how to create this sodium ion influx, we get advice from David Drew, a professor in biochemistry at Stockholm University. Influenced by his input, we came to the conclusion to use the light-driven channel rhodopsin rather than an ion channel driven by osmosis, since this could interfere with our chloride influx. Halorhodopsin and channel rhodopsin are alike in multiple ways. In addition to them both being activated by light, they both require all-trans retinol to be bound to them. This means that access to all-trans retinol is a limiting factor for the chloride and sodium ion influx that we strive for. All-trans retinol is formed when beta-carotene is cleaved by beta-carotene 15-15' dioxygenase. For this reason, the third part in our desalination system creates an excess expression of the enzyme beta-carotene 15-15' dioxygenase. This way, more beta-carotene can be cleaved so that the chloride sodium ion influx is not constrained by lack of all-trans retinol. These three parts together create a system that moves chloride and sodium ions into the phototrophic organisms. However, a threat to the solution is that osmosis causes water to follow the ions into the cells to the point where they disrupt. To evade this from happening, we designed a fourth part containing the non-selective large conductance mechanosensitive ion channel MSCL. MSCL regulates osmotic pressure, and its purpose in our system is to stop cell disruption from occurring. Before MSCL is induced and sodium and chloride ions are released back out into the surrounding water, we wanted to design a part that allows us to separate the organisms from the desalinated water. Let's move to the separation part of the cycle and explain how we solved this. We considered various ways of solving this, and ultimately we were inspired by our own Linköping team from 2019. They worked with a carbohydrate binding domain, or CBD for short, and exploited its high affinity towards cellulose. So, the CBD part we designed for our separation to be functional contains a fusion protein, separated by a linker. The fusion protein is expressed on the surface of the phototrophic organism. Since we chose to work with Synechocystis, the membrane-bound part of it is an S-layer protein called SLR1272. This part of the fusion protein can easily be replaced by another membrane-bound protein, compatible with another phototrophic organism. The other part of the fusion protein is CBD. Since CBD binds so strongly to cellulose, the phototrophs can be separated from the desalinated water by passing it through a cellulose filter. The cells bind to the cellulose filter via the CBD on their surfaces and are thereby separated from the water. After this separation step, the filtered desalinated water is ready to be used in agriculture. To summarize the desalination and separation step of the CyaSalt cycle, the modified phototrophic organisms import extracellular chloride and sodium ions, so the extracellular salt concentration is lowered. Then they can be separated from the desalinated water by a cellulose filter. The whole process is exclusively fueled by sunlight and the nutrients present in the water.
But what phototrophic organism is optimal to implement our CyaSalt desalination system in? Let's go more in depth into the cultivation part of the cycle. As mentioned, we aspire to apply the CyaSalt desalination system to phototrophic organisms. One of the reasons for this is that the photosynthetic properties of phototrophs can be exploited so that sunlight is used as the sole energy source. This energy-efficient desalination method is both economically and environmentally sustainable, making fresh water accessible to all parts of the world independent of economic standpoint. Through discussions with Sara Ahmed, who holds a PhD in environmental sociology and is on the board for WaterAid India, we got confirmation that our solution has a great potential to improve the lives of many people in affected areas. So, in order to study the cultivation of these phototrophic organisms in seawater, our project included extensive wet and dry lab studies of the growth of one specific halophilic phototroph, Synechocystis sp. PCC 6803. We acquired cultures of Synechocystis from a research group at Science for Life laboratories in Stockholm. the associate professor of that research group, Paul Hudson, also came with valuable input regarding the light conditions to use during cultivation. We did not have any prior experience of working with Synechocystis, a bacterium with a very slow growth rate compared to the doubling time of 20 minutes for E. coli. So, to optimize growth conditions, we did extensive research on what media composition to use and which external factors to adjust. We reached out to several experts for advice and guidance. Dr. Vamsi Moparthy, a research engineer with substantial experience of working with Synechocystis, was one of them. He provided us with cultivation protocols that led us to use the growth media BG11. After discussing with Julie Zedler, a junior professor in synthetic biology based out of Germany, we also started growing the bacteria with open atmosphere access. We did this by sealing the culture flask with cotton plugs, and it resulted in a considerable increase in growth rate. But since our desalination system ultimately would require Synechocystis to grow in seawater, we retrieved water samples from the Baltic Sea as well as North Sea. We studied the growth of Synechocystis in both, using BG11 as a control. The results can be seen in this graph, and they clearly exhibit substantial growth in BG11. In the non-autoclaved samples, however, the growth plateaued after only 5 days, after which the Synechocystis began to die. After determining this and analyzing the dying cultures, we found various other phototrophic organisms still thriving in our seawater samples. We were able to speculate some of the species by studying the samples under a microscope. This led us to the conclusion that there are probably phototrophic organisms other than Synechocystis that are more suitable to implement our desalination system in. The collected data from our cultivation experiments was applied to a model that we created to predict the growth of phototrophs under different conditions. When applying our data to the model, we were able to see the effects of different growth conditions. Furthermore, this model was designed in a manner so that any bacteria can be analyzed depending on the input data that is provided. This means that potentially the growth of both modified Synechocystis as well as other phototrophic organisms found in our seawater samples can be studied.
Considering the future, real-world implementation of our project, as well as its environmental implications and social effects, is something that has been central to us throughout our whole project. This led us to consult Dr. Sara Ahmed, who, among many other things, is on the board for WaterAid India and Wetlands International South Asia. She informed us of the water scarcity in India and stressed the need for economic availability and environmental sustainability in future desalination methods. The search for further insight in the requirements for a method brought us to the Swedish island of Gotland, situated in the Baltic Sea. Gotland is one of the few places in Sweden where water scarcity is a real, very present problem. On Gotland, we met with 4 farmers for in-depth discussions about how they are affected by water scarcity. We also consulted them regarding how we should design our upscale desalination system. All of them had experienced hardships caused by the lack of fresh water. They told us that due to droughts on the island, many farmers have had to ration their water and ultimately let substantial amounts of crops dry out and die. This has largely affected the farmers' economies, and with the summer temperatures increasing year by year, water scarcity is only getting more severe. Watering bans have become more common, and in 2018, when Sweden experienced an exceptionally warm summer, some farmers who usually grow their own cattle fodder had to buy it from the mainland instead, costing them an extra 60,000 to 70,000 euros. The financial effects of this can still be felt to this day for many of the farmers. According to Rune, one of the farmers that we met, many farmers are worried about their water supply running out. When presented with our idea of using genetically modified organisms as a solution to their lack of fresh water, the consensus from all 4 farmers was that as long as it has been tested and approved by the government and other institutions, they would consider using our product. However, they all agreed that the biggest concerns for them is that it would need to be a small-scale, cost-effective setup that could be implemented locally on each farm. Let's take a look at what we came up with. Our industrial setup would utilize gravity as well as Pascal's law of communicating vessels. This way, we would achieve an eco-friendly approach by reducing energy consumption and thereby desalination costs. A channel would lead the seawater into tank number 1 and fill it until it is at the same level as the sea. The modified phototrophic organisms would be added to tank number 1, where they would be cultivated with nutrition from the seawater in addition to sunlight. Desalinated water would be transferred from tank number 1 to tank number 2 by a pipe placed close to the water surface containing multiple filters to capture the modified organisms. Desalinated water, ready for agricultural use, would then be pumped out through tank number 2 by a solar panel-driven pump. The waste biomaterials of phototrophic organisms precipitated to the bottom of tank number 1 would be led by another pipe to a waste tank equipped with a gate that the farmers could access when needed. Here, the biomass would dry out, preparing it for biofuel production. Considering that the farmers told us that they wouldn't need their total daily water consumption to be provided by our solution, a smaller tank size could be used depending on the desired water amount. Furthermore, we made an approximate calculation of the expected costs of our upscale desalination method. The farmers explicitly told us that today they pay between 0.4 and 0.6 euro for 1 cubic meter of water. They said that for our method to be financially defensible for them, our price would have to be lower than that. From this, we obtained a water price of approximately 0.3 euro for 1 cubic meter of water. However, if only considering the operating costs, the price for 1 cubic meter would be as little as 0.002 euro. Compared to what the farmers pay today, these prices are considerably lower and qualify for what the farmers presented as a requirement. However, due to the lack of experimental data, most aspects of our industrial setup are based on assumptions. Nonetheless, based on our estimations, we have established that CyaSalt could provide a cost-efficient desalination method that succeeds the requests made by the farmers we met and interacted with. Furthermore, it's a desalination method that is not only applicable in Sweden, but that could be adjusted to work for coastal farmers anywhere in the world.
In conclusion, our project CyaSalt has laid the fundamental groundwork for future research. We have designed a part collection that creates a system with the purpose of enabling phototrophic organisms to efficiently desalinate seawater. In addition to desalinating the water, our system also includes a solution for separating the modified organisms from the water after desalination. Furthermore, we have obtained critical results from our extensive experimental work with Synecocystis sp. PCC 6803, as well as from our model. These results impacted the future visions for our project and will also aid future research on deciding which phototrophic organisms to ultimately integrate the CyaSalt system in. Moreover, we have created a design for the scaled-up version of our desalination method. This design presents a cost-efficient desalination method that would make freshwater for agricultural use substantially more economically available than it is today. And not to forget, the CyaSalt desalination process would rely solely on sunlight as its driving energy source, meaning that it would leave no environmental footprints. Advice and input from various people has greatly impacted and shaped our project into its current state and design. Whether their expertise has been on the agricultural industry, Synecocystis sp. PCC 6803, ion channels, or other crucial aspects related to our project, we want to extend a special thank you. Moving forward, us in the Linköping iGEM team 2021 are eager to see CyaSalt evolve further and reach its full potential, so that we can hopefully offer a solution to the global freshwater crisis. CyaSalt!